So our next speaker is Dr. Lily Wang. Lily Wang, she's a PhD research instructor in the Department of Medicine at the Vanderbilt University Medical Center, where she investigates the arrhythmia mechanisms underlying genetic cardiac disorders. Her research centers on how metabolic stress disrupts cardiac electrophysiology using human-induced PSC-derived cardiomyocytes models to dissect disease mechanism at the cellular level. She will be presenting her work on how metabolic crisis drives QT prolongation in TANGO2 Deficiency Disorder.
Hi, good afternoon, everyone, and thanks for Lina's kind of the introduction. And also, I want to thank the foundation for giving me this great opportunity for sharing my work. So my talk will bring you guys into the heart very specifically. So we just wanted to understand why metabolic crisis cause the QT prolongation in TANGO2 deficiency disorder. No disclosure, and this study is funded by the TANGO2 Research Foundation. Okay, let's back to the science. So actually, our TANGO2 study is beginning in 2019. Dr. Prince Canacreel in our center had a patient who had a severe QT prolongation and also no. Okay, so forget. Has arrhythmia, so the patient actually responded really well to the L-type calcium channel blocker Verapamil. So later, the genetic test shows the patient actually carries the TANGO2 exon 3 to 9 deletions. And so most of you already know what TANGO2 do. It's just to help the mitochondria function and maintain the Golgi organization. And also in the TANGO2 deficiency disorder, the patient have the metabolic crisis due to impaired the non-chain fatty acids oxidation. And during the crisis, patient has severe QT prolongation and ventricular arrhythmia. So we want to understand how metabolic crisis driving the QT prolongation in the patient. So we just generate the TANGO2 knockout iPS cells and differentiated the control and the TANGO2 knockout cells into our myocyte. So the Western blot just confirms there was no TANGO2 protein expressed in our knockout line. Our first question is, does this TANGO2 knockout cells present or have the disease phenotypes? To answer this question, firstly, we use the Pencilville HR. This is a biosensor can measure the cellular ATP over ADP ratios. And we treat we challenge these iPS cells under the different kind of the few conditions. So firstly, we tried the glucose. And 5.6 millimolar glucose is a physiologic glucose concentration in human. So you can see in the glucose conditions, ATP over ADP ratio does not change in TANGO2 knockout cells. Next, palmitate. Palmitate is a non-chain fatty acid, is predominant of non-chain fatty acids in human. If we challenge our iPS cells with this 0.1 millimolar palmitate, what happens is ATP over ADP ratio significantly decreased in the knockout cells. And since patient always has experienced the severe crisis after the fasting, so we culture our iPS myocyte in the glucose, pyruvate, and glutamine-free mediums for 24 hours to challenge these cells, and then treat with 0.1 millimolar palmitate. You can see ATP over ATP ratio further drops in our knockout cells. So this data suggests palmitate actually caused the ATP deficiency or metabolic crisis in the TANGO2 knockout cell. So we ask why palmitate did this stuff. So we use the Seahorse assays actually look at the mitochondrial functions. We measured the mitochondrial oxygen consumption rate, OCA, and also calculated ATP production rate. So you can see OCA and ATP production rate were unchanged in TANGO2 knockout cells. under glucose. So this suggests glucose actually TANGO2 deficiency or loss of the TANGO2 did not affect the glucose oxidation in the iPS myocyte. Nextly, palmitate. So OCA and ATP production both significantly reduced in the TANGO2 knockout cells. So this data suggests loss of the TANGO2 actually impaired palmitate oxidation in TANGO2 knockout cells. How about the palmitate post-fasting? The result is really interesting. So look at the control groups. You can see palmitate post-fasting actually increased the OCA, but did not change the ATP production rate. Look at the TANGO2 knockout cells. That's the different story. So OCA did not change post-fasting, but ATP production rate significantly decreased in the knockout cells. So this data suggests actually fasting worse the ATP deficiency or worse metabolic crisis in TANGO2 knockout cells. And clinically, patient always has beneficial from the vitamin B treatment. So we are asking, like, can we just reproduce the beneficial effect of vitamin B in our iPS models? So we treat ourselves with vitamin B5 and B9 for 2 weeks. The reason we pick the B5 and the B9 because they are important for the CoA synthesis, and the CoA is important for the palmitate oxidation. So you can look at the data here is like compared to B5 or B9 individual treatment, combined B5 and B9 significantly increased OCA and ATP production rate just to boost the palmitate oxidation. Because of the increased ATP production rate, you can also see like ATP over ADP ratios actually restored in TANGO2 knockout cells. So this data suggests, B5 and B9 just improve the palmitate oxidation upstreamly in the TANGO2 knockout cells. So stop in here, we're just asking next question. Does this TANGO2 kind of the palmitate oxidation defect is due to kind of the broadly disrupt the cardiac structure or function? So we did RNA sequence and we compared under 2 conditions. One condition is glucose. Everything was fine for TANGO2. And for the palmitate post-fasting, we see most severe phenotype. You can see the gene related to contraction, ion channel, calcium handling, or mitochondria. Everything was fine in TANGO2, except for one gene. Oh, seems like it's just a kind of the block. Okay, so except for one gene, CPT1B. CPT1B, this is kind of the enzyme just to transport the fatty acids into the mitochondria. This gene is significantly increased. So this can explain why we saw the palmitate oxidation decrease in the TANGO2 knockout the cells. Cell just wanted to compensate this. They want to transport more palmitate into the cell, into the mitochondria, and increase the palmitate oxidation. So based on this result, we know TANGO2 deficiency actually does not affect the cardiac structure or functions, but specifically impairs the palmitate oxidation. So, and at this point, we know the TANGO2 knockout iPS models have like the metabolic defect. We're asked the next question, can this defect actually drive or changes the electrical behaviors in these TANGO2 knockout cells? So we use the like extracellular field potential. So this extracellular field potential is like the EKG like recordings from the cardiomyocyte in a dish. And we optically paste these cells to control their beating rate. So the difference we observed were not simply just due to the different beating rate. And we compared the field potential duration, FPD. So field potential is like how long the electric signal just lasts during each beat. So you can see under the glucose condition, TANGO2 knockout cells behave normally. FPD is fine. But once we treat these cells with palmitate, only just the palmitate, you see FPD gets prolonged. So it means like the cells, just the electrical signal lasts longer to back to the normal during the each beats. And the palmitate post-fasting, the FPD prolongation gets more severe compared to the palmitate only. And also we observed like the palmitate, under the palmitate stress, the FPD alternance gets increased. FPD alternance is kind of the beast-to-beast instability. So you can see one non kind of the FPD and coming with a shorter one. So this is kind of the warning sign for the arrhythmia risk. And the EFP data just tell us like, yeah, the TANGO2 knockout cells take longer, just like back to the have the longer electric activity during the each beat. But we do not know what exactly happens in the individual cardiomyocyte. So nextly, we just use patch clamp to measure the single cell action potential duration. So this single cell action potential duration just tell us like the electric activity during each kind of the beats. So you can see consistently with FPD data, glucose TANGO2 knockout cells is fine. Palmitate and palmitate post-fasting actually prolonged action potentials. And action potential alternance is also the base-to-base alternance is kind of instability, just like the changes from each kind of the action potential. So TANGO2 knockout cells, you can see they have the one long base and coming with a shorter base, so non-short pattern. And palmitate and palmitate post-fasting both increase this alternance. So here we know, yeah, the TANGO2 loss of the TANGO2 cause the ATP deficiency and prolonged action potential and increase the alternance. So does this ATP deficiency actually drives the action potential prolongation in this TANGO2 knockout cells? To answer that, we just deliver the magnesium ATP directly into the cells while by our patch pipette. You can see magnesium ATP deliver actually prevent action potential prolongation and alternance in TANGO2 knockout cells. So this data suggests the action potential prolongation observed in TANGO2 knockout cells is ATP dependent. So here we know the TANGO2 loss of the TANGO2 just impairs the palmitate oxidation, cause ATP deficiency, and cause action potential prolongation and alternance, and increase arrhythmia risk. If we fast these cells, actually makes the situation worse. So palmitate oxidation further reduced, and the metabolic crisis gets worse. And combined B5 and the B9 treatment actually can restore the upstream the palmitate oxidations in this IPS myocyte. Okay, next. Clinically, our patient respond really well to the verapamil is L-type calcium blocker. So we ask, can we reproduce this effect in our dish? So we acutely treat the verapamil to our IPS myocyte. You can see after acute verapamil treatment actually prevent the action potential prolongation and alternance. Because verapamil is L-type calcium blocker, where just the most obvious explanation is TANGO2 would be TANGO2 knockout cells increase L-type calcium current, and so verapamil has this protective effect. So we directly measure the L-type calcium current and find out there is no difference in L-type calcium current between the control and the TANGO2 knockout cells. Okay, so and next possibility explanation will be maybe TANGO2, maybe palmitate stress changes the intracellular calcium handling in the cardiomyocyte. So verapamil just block the calcium influx and have the protection. So my colleagues Kwang Su Kim just measure intracellular calcium present and find out, under the permitted stress, intracellular the TANGO2 knockout cells have abnormal calcium handlings. And if we give the vapor treatment, vapor correct everything. And our next question is, does vapor do something to the permitted oxidation? So we did the seahorse assays, and we find out vapor did not correct permitted oxidation in TANGO2 knockout myocyte. But but vapor actually correct intracellular ATP over ADP ratios in the permitted conditions, because vapor actually is reduced the working kind of energy burden to preserve the energy standards in this cardiomyocyte. So from this part, we know TANGO2 deficiency cause ATP deficiency, cause the calcium mishandling, and cause the action potential prolongation. And if we give the vapor can block the calcium mishandling and rescue the action potential prolongations. So this also prevent our IPS model can use as kind of the real model to understand what happened in the TANGO2 knockout or happened to TANGO2 deficiency disorder. But we still have one missing link. We want to try to find out a downstream ion channel who can collect the ATP deficiency and the calcium mishandling together, driving the action potential prolongation. So we did a lot of the search, and finally we found a Y-ion channel that called the TRPM4. This channel actually is high expressed in the human heart. This channel is regulated by the ATP and the calcium. So you can see ATP can inhibit this channel, calcium can activate that. And this channel is increased expression in the TANGO2 knockout cells. And once we use siRNA mediate this channel knockdown, what happens to action potential? You see action potential gets prevent the prolongation. And so our next question is, this channel kind of interact with the metabolic defect in the TANGO2 knockout cells? We did the seahorse assays and find out TRPM4 is downstream of the metabolic defect. It knocked down the TRPM4 did nothing to the permitted oxidation, nothing to the ATP production rate, nothing to the energy standards. So and TANGO2 kind of the phenotype still there, but it can rescue the arrhythmia. So here we know TANGO2 deficiency cause ATP deficiency, cause the calcium mishandling, and cause action potential duration, and because it's activated the TRPM4. Okay, so from our study, we learned that TANGO2 deficiency actually impaired the fatty acids oxidations and reduced ATP over ADP ratios in the cardiomyocyte, and activate the calcium. So then activate the TRPM4, prolonged action potential duration, and increase arrhythmia risk. Combined B5 and B9 treatment actually can restore the permitted oxidation, and the vapor can reduce the calcium-induced the stress and inhibit the TRPM4 activation and rescue the action potential prolongation. So this kind of our study explain why the patient can respond really well to the vitamin B complex supplementation and also the vapor, because vitamin B complex supplementation rescues the upstream in defect, and vapor is downstream in fact. And the TRPM4 can be the therapeutic kind of the target for this metabolic crisis induced arrhythmia in TANGO2 deficiency disorder. Okay, so for this study, I want to thank my mentor, Dr. Beyond Noman, and all my colleagues. And also, I want to thank Sam and Dr. Brooks for their help in this study. Thank you so much. Happy to take any questions.
Thank you, Dr. Wang. Questions?
Lily, that was really cool data. I have a couple of different questions. Have you tested this in other fatty acid oxidation disorders like VLCAD to see if you have the same phenotype of changes in TRPM4 in response, like sensitivity to calcium channel blockers? And if it's really driven by a decrease in ATP availability in cardiomyocytes during stress states, can it be a more universal treatment for patients with fatty acid oxidation disorders using calcium channel blockers? And then, have you tried other fatty acid oxidation disorder treatments like C7 or triheptanoin to see if you have response in the same phenotype?
For the first question, for other fatty acid disorder, we're going to do that. We're also very curious how ATP deficiency and this calcium mishandling, this also just a universal kind of the phenomenon in the other kind of the disorder. For the second question, you ask the other drugs.
So other drugs that are used that long-chain fatty acid oxidation disorders like triheptanoin or C7, like can you get a similar sort of rescue? Because if it's really driven by just decreased ATP availability, if you provide other substrates, in this case you're using palmitate as your only energy source, and if you have loss of CPT1B, you're not going to import it. But if you have other things that are able to get in there that exclude or bypass that pathway, and you have energy production and energy maintenance, can you avoid a lot of these long-term like defects?
That's great, really great question. We did not try that, but I think will be our next project.
Other questions for Dr. Wang?
Hey, Lily, great talk. Great job that you guys are doing. Did you try to separate out, did you do experiments looking at B5 and B9 alone? And did you look to see if B5 and B9 rescue the APD?
We did not actually start the B5 or B9 on the EFP or APD. We just want to see like this kind of the B5 or B9 or combined this treatment actually helps with a metabolic crisis. So based on our data suggest if you correct metabolic crisis in TANGO2 knockout cells, actually you can prevent action potential kind of the prolongations.